So my topics is to introduce you some of the main concepts based on fluorescence. I hope or I think that many of you already played a little bit with, let's say, with microscopy. Oh, I'm sorry. Okay. But I just want to remind, because we all take advantage of jobs, of work done by people before us. And let us just remember that the fluorescence phenomena was discovered several years, even centuries ago, but not immediately, let's say, exploited and not immediately used for our own purpose today. Among the different names, you will have this presentation, so I will not read or whatever is written all the time. I just want to remind one name, because this Sir George Gabriele Stokes, I enjoy his name not only because it's like my name Gabriel, but because Stokes, you will heard a lot of time the name Stokes shift in microscopy in fluorescence, and so it's important to know that his discovery was made in the 19th century. One in the 1843, he described for the first time what he called a luminescence. In fact, we know now that it's not correct to call it luminescence. It's a fluorescence phenomena, showing that when you illuminate with a particular light a compound, in that case was calcium fluoride, these molecules are able to produce a light with a different wavelength, a different color in our case. For this was the main base, the starting base of the theory of fluorescence that was furthermore developed by physicists, by Joseph von Lommel, in the 1875, then showed the first time how really a substance is able to absorb light and emit a new light as fluorescence. This is another small slide showing, let's say, a little bit more near our own world, because here we are also entering in what is the microscopy path, because the first time was completely, let's say, separated from the microscopy, while here people start also showing the excitation spectrum of the dyes, the phenomena of fluorescence quenching. During my presentation, I will give you an idea of what these concepts are, the polarization of dyes, and all the history of what we know now of fluorescence. One of the main points that another name that you will definitely heard a lot during this presentation and probably also during future presentation is Jablonski, which was the physicist that create what is called now Jablonski diagram. But when I thought about starting asking myself, because probably we all thought about this, why we need fluorescent microscopy. One of the main ideas is that we all played a little bit with a different microscope. We know bright field microscopy, and one of the main problems that we probably met with our own bright field microscopy is that on all type of microscope, what we saw into the cells is not fully distinguishable. We maybe now know what we are looking at, but there is no, let's say, no signature of what we are looking at. So this is a DIC image in bright field microscopy, but we probably know as biologists or as scientists that this is a nuclei, that this is the edge of the membrane. These are probably some sort of vesicular structure, but we really don't have a signature of what we are looking at. So why we need fluorescent microscopy? First of all, because we are able to have a higher resolution than comparing using the full spectra of light given by white light, because we have an incredibly high contrast, as you can perfectly understand. Working with something bright on a black background gave us a high contrast. Contrast is one of the main limitations in microscopy. Everybody thinks about magnification as a main limitation in microscopy, but after all, magnification is physically limited by the, let's say, by the law of physics. You will heard also people in this course going over this law of physics using super resolution microscopy. But magnification is a sort of fixed limit that is not really easy to go through. While contrast is the main concept of microscopy. All techniques in microscopy, both bright field and fluorescent microscopy, have as an aim to achieve, the main aim is to achieve the highest possible contrast, because allow us to better describe what we are looking at. Another element that is specific of fluorescent microscopy is the high specificity. As you can see here, you perfectly understand that the blue color is something's different from the green color and something's different from the red color. We are looking through three different structures, three different biological structures with their own function: nuclei, actin filament, microtubules, and so on. So we are really able to have a high specificity. Another important features that right now we know that we can explore with fluorescence, and we heard about this tomorrow. We will hear tomorrow using in the course of quantitative imaging, is that we can even be quantitative in what we are looking at, because the fluorescence is an active process. So the light emitted is somehow correlated with the quantity of what we are looking at. Finally, one of the main features with one of the main advantages of fluorescence is that we can also look in live cell imaging. So the image we look at right before, I'm sorry, just going back one slide, this image, what we can achieve on this image if we are looking with fluorescence. Using three different dyes, we are able to see the same image as before with three different colors, each one telling us actin, DNA, and vesicular structure, and giving us this kind of image. This kind of feature with high contrast, high specificity is like a summary of what we can achieve with fluorescence, giving us a lot more information than what we are generally able to achieve with a classical bright field microscope. So going to what is fluorescent principle, we can summarize a little bit saying that what is fluorescence? When we are illuminating a compound with an energy, with a light, this light is also have its own energy, this compound is able to emit a different light with a different level of energy, so to a longer wavelength. This phenomenon is called as fluorescence. I will probably repeat this concept few times, even more times, even many times, so don't be bored about this concept. Often, we will hear about this in the afternoon, our specimens have their own ability to produce fluorescence. This is called autofluorescence. Almost all molecules have their own ability to respond in such a way. So we also need to consider this from a technical point of view. The key features in the fluorescence microscopy is that we are not using a transmitted light, but we are eating our compounds with the light, and then we are collecting the light produced by our sample. So we are not measuring, like in bright field, the light passed through the sample, so the light absorbed or the light reflected or deflected by our compound. We are measuring the light produced by our samples. In this case, just as another example of specificity, you can see mitochondria, you can see tubules, you can see DNA, all perfectly stained in microscopy. This is just, as you probably understand, I'm quite passionate about history of microscopy, and that's why you see this kind of machine. You will not, I think, play with this machine during this course, I hope, but this is just an example of one of the first microscope. You see here let's say the classical compound microscope stage, and all this part is the one allowing us with this filter barrel to produce a single wavelength able to stimulate our fluorophore and to produce our own fluorescence. This was produced during the 30s, this machine, and from, I think, that moment from Zeiss and Jena. So, but in a molecule, what really gave us a light as an answer? What gave us fluorescence? in this small drawing, you probably all have a chemical background, an atom, an electron, or several electrons, a quantum of energy given by light hitting our molecule, this electron making a jump, going in what is so-called an excited state, and going back from this excited state, releasing newly produced energy. This is what I mentioned you before, was one of the Stokes observation. When you have an absorption of the light, the electron going to an excited state, and then the electron not staying stable in this an excited state needs to go back to the zone basal state, releasing light in this way. So this is what I called before, what is called the Jablonski diagram. And you can see in this kind of diagram that all the electrons in the basal state have their own basal state of energy, and then receiving energy, they jump on the excited state. This kind of condition that is perfectly described by this diagram is at the basis of all type of fluorescent microscopy. I just want you to remind that going up to the high state, you have a vibrational state here producing heat generally, producing a dispersion of energy, and then you have the emission of light going back to the basal state. This kind of vibrational state is what is the vibrational state of emitting heat, is what is generally cause the reduction of our efficiency in the fluorescence, and is also what generally cause the loss of fluorescence in our samples, because sooner or later this kind of vibrational state breaks the molecule we are studying at. So this is just a small reminder, because I often receive this kind of question about the difference between fluorescence and phosphorescence. We heard about these two phenomena, and I think that they can be better described with the Jablonski diagram. That is because they are both types of luminescence produced by the molecule. But in fluorescence, the emission of light is extremely, extremely fast. We are speaking about nanoseconds as a time scale. We have a rapidly produced energy right after the absorption, while the phosphorescence emission is a completely different state, where when we have the emission that can last milliseconds or even seconds or even minutes. Let's think about the phosphorescence of your watches. So summarizing what we have in fluorescence, phosphorescence, and the also so-called delayed fluorescence. In the first case, we have light hitting the electron going in the excited state, vibrational energy release, and then the real quantum of energy release as a light. In phosphorescence, the vibrational state goes to a transition state where electron can stay much longer, and that's why they can produce light for a much longer time. And then we also have the transition state of the triplet, that is so-called the delayed fluorescence. All these concepts that on a regular epifluorescence microscope can be a little bit tricky to summarize and to think about how useful they can be, became quite clear when you move from a classical epifluorescence microscope, let's say standard fluorescence microscopy, to the newly developed super resolution microscopy. Because in several cases, the different kind of fluorescent microscopy you will heard about during these days and in your career, they all exploit different part of the Jablonski diagram. In a regular epifluorescence microscope, we simply take advantage of the fluorescence rapidly produced by the molecule, but we need, while with working with a storm, a palm, a super resolution microscope, we also work using the excited triplet state. What I want you to focus in this kind of diagram is that fluorescence is extremely, extremely fast, but is even, and one of the microscopy in fluorescence also used is the FLIM microscopy, where you study the lifetime of this fluorescence. So you really can have a plethora of techniques working each one of a different part of the phenomena of the fluorescence. So you can work with lifetime, you can work of excitability, you can work on the triplet state or the basal state of the fluorescence. You can work on the ability of a molecule to be quenched, and so on. So consider that this kind of phenomena is extremely complex, but each technique tends to be quite focused in studying one of these phenomena. We have, just as a reminder, we have a lot of natural fluorescent compounds in nature, and this is how they were discovered. The fluorescent phenomena here we have. Oh, I'm sorry. We have an example of a molecule with its own excitation spectra and its own emission spectrum. We will see in a few minutes what we mean for excitation and emission spectra. This is just to remind you that we are plenty in nature of fluorescent molecule, and even, okay, let's say the Nobel Prize was won discovering, or at least cloning, the green fluorescent protein, which is a natural protein present in a particular kind of jellyfish. We have right now the ability of use several of these natural compound present in literature or chemically developed and specialized probe to be able to detect either protein-protein interaction, DNA-protein interaction, and even enzyme, as I mentioned before, enzyme activity. So we really can have a plethora of different techniques that can be used with fluorescence. One of the things that I think people working with fluorescent microscopy likes more, as you saw in the image before, is using several different colors. So several different fluorescent probes or fluorescent dye. How we can do this? We can do this because each fluorescent molecule have its own ability to respond to a particular wavelength and to emit at a particular wavelength. In this case, I just place here this cover from Journal of Cell Science, where we have 5 different fluorescent proteins, all coming, in fact, from the GFP, the starting, and then the other one was modified from this. And this protein, you can see the left line is every time the excitation spectra is the ability of, this gave you the idea of the ability of a different wavelength, reported here, to excite the molecule, while the second line here in green is showing you the emission of the photons from this molecule, from this, in this case, from this fluorescent protein. So here, and you just see how GFP has its own peak of excitation here, and really a small distance, a small Stokes shift, we have its own peak of excitation. While each one of our possible fluorescent protein has its own fluorescent, called fluorescent spectra. And here you see just the emission reported here. So going in a little bit in detail on what is the emission excitation and emission spectra, because you will need to know which kind of probe or which kind of fluorescent protein you can use together, and and you will see in the next presentation how to set up a system based also on this information. So as already mentioned, every fluorescent molecule has its own absorption spectra, here reported as a profile, and its own emission spectra profile. there is generally a sort of overlap. This is just to let you know that it's not an all or nothing phenomenon. Each molecule is, here is the percentage, sorry, is a normalized percentage. So we know that this molecule is 100%, or at least at the maximum of its ability, excited at about 550 nanometers. So let's say a green light, green-yellow light. But if you see E-zone absorption spectra, this phenomenon is not an all or nothing. Even already at 500 nanometer, we have at least 20% of this fluorescence that is achieved. So this is important because when you work with a fluorescent dye, you have to understand that you are never using all of its potential, and you have to take care of the different ability of the different fluorescent dyes. This kind of absorption spectra, or ability of the electron to be, let's say, switched to excited state, also gave us an emission spectra. So even the same electron going back to the basal state producing light, and this light, this amount, this quantum of energy released, is released, let's say, in this case, preferentially in this yellow light at about 570 nanometers. But in fact, also this phenomena is not an all or nothing, and is not specifically to this wavelength. We also have a long tail, short or long, depends on your fluorophore, describing that this molecule still produces 40% of its light over 600 nanometer. So we have what is so-called the spectra. So we have a huge amount of light and a huge difference between the starting and the end of the spectra of this molecule. The distance between the peak of absorption and the peak of excitation is what is called Stokes shift. If you remember the name of the first scientist I mentioned, this is one of his main contributions. It was the first one describing that you have this kind of movement to a more energetic wavelength to a less energetic, longer wavelength. I tend to stress a lot because it's quite easy to mix up and thinking that longer is higher energy. No, longer wavelength is lower energy, and that's why we have this kind of phenomenon. We lose, why low energy? We lose energy in vibrational states. So we will never be able to produce light to a high, or to produce higher energy than what we gave to the molecule. So this small spectra, in fact, is a sort of sum of several different physically encoded aspects of the molecule. I will not stress a lot on this, also because I'm much more enrolled in neuroscience. I really enjoy working with fluorescence, but I'm not a physicist or a chemical expert in all this kind of aspect. But just to remind you that what you just saw as a spectra show you the absorption coefficient that you have at the peak of absorption. You can also understand and calculate the quantum efficiency, so the ability of each single molecule to produce back your own light. You should consider that generally the fluorescent phenomena is quite, let's say, is quite inefficient, or let's even say is extremely inefficient. You need to use a huge amount of light to make molecule producing the small light you need to describe your sample. That's also why you generally need, after all, to use this kind of beautiful, expensive, and extremely powerful cameras, because the amount of light produced by your sample in the fluorescent phenomena is extremely inefficient. You really have a low amount. But you need to know how inefficient or how efficient is this phenomenon. You need to know the Stokes shift, so you need to know the characteristic of your fluorophore, and in this way the width and the height of your spectra. Is generally are described as a mirror image rule. Is not in real life. This almost never happen. You never really have, let's say, the absorption and the emission completely specular, completely as in a mirror. but generally describe quite well that if you have a really large excitation spectra, you probably also have a really large emission spectra. And this should give you some details about how we use this fluorophore. Now a lot of companies are really working hard or worked really hard in the past years to produce extremely narrow excitation and emission spectra, because this will allow us as scientists to use more color, more molecule, and so to stain more specific organelles, structures in cells, tissue in once. So that's why one spectra like this right now tends to be narrow as possible. We have, in this case, a real emission and excitation spectra. This is with a so-called high Stokes shift, and you see that the absorption generally also have several different peaks, because these are described by the different electron that can be excited, while here you have generally a single emission spectra at the end. But this is, again, quite large, so you can decide to see them in several different states of light. One of the characteristics that I think we should remember about the fluorescence emission is that even if we have several different excitation spectra, we have, after all, one emission spectra. I don't know how many of you use the nuclear dye, like DAPI, UXT, or this kind of intercalating dye. These are generally structured to have several different excitation spectra, ranging from the UV light to the blue light, while their emission spectra is one, and is, in our case, in the case of DAPI or in the UXT, is in the blue light. So you should consider that even a completely different wavelength will give you a different emission spectra. So it's not that exciting in one wavelength will give you, I don't know, an emission here. The emission spectra is all defined by the molecule itself. So this is just a couple of examples of what we can achieve with different fluorophore and with the superimposition of the different image we collect. These are collected from the web, from Nikon and other microscopy producer websites. So here we have different probes specific for nuclei, cytoskeleton, and actin and pallidine staining. So a small reminder before having a pause is that we have why we really want fluorescent microscopy. We need high resolution for what we want to see. We definitely can achieve a high contrast because we can see light on a black background. We can be highly specific, and we can be quantitative because the phenomena described by the Jablonski diagram is a quantitative phenomenon. And we can work in live cell imaging, and you will probably heard about this in the future days. As I mentioned you in the Jablonski diagram, the excitation is extremely fast. It's about 10 to minus 15 seconds. So we are in the femtosecond scale, while the emission is slightly longer. So we can go from in the range of nanosecond. And describing how we can exploit this phenomena in our sample. I will be really fast, and I will go really through this kind of description because you will heard from the technical point of view from Giacomo, really the description of how the microscope in this case works. But if we think we need to stimulate our molecule with a wavelength and to collect the light produced by this molecule. So it's clear that while in a regular microscope we use a regular white lamp, in a fluorescent microscope, given the inefficiency of the fluorescence produced, we need particular kind of lamp, specifically able to produce its own spectrum of excitation. Right now, we also can take advantage of the usage of LED, LEDs, with their own emission spectra, with their own illuminating spectra, but I have to admit that right now the other lamp, mercury lamp, xenon lamp, metal halide lamp are still the more used. These are extremely powerful lamp and illuminating with specific wavelength our sample. This just to, you probably already know how a microscope is done, but just what I was stressing before is that in a regular bright field microscope, the light needs to pass through the sample, through the optics, and then reach either your eyes, either a camera. In a fluorescent microscope, is your sample producing light, so you don't want to be blinded by the light to illuminate the sample. And that's why the light, starting from the upper part in this case, pass through the optics, so is also focused on your sample, and let's say is going away, the light you used. And then what are you collecting? You are collecting the light produced by your sample. This is the main difference in a microscopy, in a fluorescent microscope comparing to the bright field microscope. So bright field microscope, white lamp, you have a lamp here, light going through here. Fluorescent microscope, you have the light that needs to be focused on your sample and going back to, not going back, sorry, and the light and going away, while the light produced is what you're looking at. To do this, and you will heard about in a few minutes after my presentation, you will need a particular kind of filter, which is generally called, in a wrong way, dichroic filter. In fact, it's not a dichroic, it's a dichromatic filter in the end. It's a filter allowing us to use a specific wavelength to stimulate our sample and to collect the specific wavelength from our sample. I will not go through in the detail because if not, the next speaker will kill me. So it's gonna be, but this is just to remind you why these filters are every time part of the microscope and why you need to know how they are made, how they are designed, and how they work. In the end, just a couple of, to relax all of us a little bit and not to be completely burned after only the first presentation. So it's just to remind you why I would also add that why we use fluorescent microscope, also because it's, I think it's beautiful. I think it's allow us to produce not only scientifically meaningful image, but also beautiful images. And so to describe the nature in a really different way and to be specific in what we are describing. And also because we are right now really, it's like having a huge amount of new techniques, all collected, all mentioned as in the whole family of fluorescent microscopy, but we can really think about directing, coupling our fluorophore with a molecule, using specific fluorescent dye. We also have fluorescent dye able to change their color upon a specific phenomena. We can produce fluorescent protein in fusion with our own protein of interest, and so follow the protein in live imaging. We can merge image from different colors and understanding how different protein works all together. So we really have a huge amount of possible techniques that we can enjoy and we can take advantage of. Going from whole, let's say, whole body or whole animal models to single cells to tissue to a specific cell. As presented before, I'm working in the University of Trieste as a neuroscientist. I'm not a technician, specifically a technician of microscope, but I really believe that, especially working in neuroscience, what you see, a shape, is a sort of the describing of the function. So as you can understand, it's essential to describe the shape and the staining of some things to know also its own function. I will give you in the presentation also some of the, let's say, dozen of possible links that you can exploit to go deeper and further in the phenomena of fluorescence, because it's really a fascinating world. For additional information, I can also give you some other details, but given the timing we have right now, I think that if you have any kind of question, we can go further with this. Please let me know. Thank you.